It's a pleasure to be here. I was here last year and expected to come to this little intimate little group and brainstorm and talk about different things. And I'm in this huge initiative, and Amber, it's really amazing what you've done in this 1 year. I'm very, very impressed and very excited about all the progress. And obviously, the connection that you're making here, UMass, we know the group very well. Esma and I have not met before, but certainly I think post this talk we'll have some very good discussions afterwards as well. Adrian Craner, as she mentioned, is actually a founder at Stoke Therapeutics. We think of ourselves as a very a company that's really focused on mRNA processing, the deep knowledge of that, and we're trying to use that to translate into drugs, basically. We are now a public company, so I have to have a disclaimer. So really, Asma has provided a lot of the background that we were just talking about. And this approach that we're talking about is particularly useful in haploinsufficiency diseases. And I'm just going to push this out. And this is really an illustration of exactly what we're talking about. In this case, the example really is in the context of SC1A, which is our lead project. But it's also a generic slide in many ways. And what you're really looking at is the ability to drive these changes in a way that's mutation independent. So in this case, there are mutations that cause loss of expression of one of the alleles, right? And in the second allele, what you have is both a productive and a non-productive message itself. And that leads to then half of the expected protein. And what we're talking about in general then is the ability to use antisense oligonucleotides to change this profile. So we're really, we are increasing as well the mutant RNA, but if it's a loss of function message, we still don't lead to obviously expression of a protein. And it's only that in this productive wild type message that we increase the level of the message and therefore the level of the protein, ideally to wild type levels, essentially. So that's the concept. So how do we do that? As we were just talking about, very, very large databases. And by these bioinformatic evaluations, then what we then look for is this universe of very small elements of sequences that we can try to find. And these could be alternative 3', 5' splice events, inclusion of exons, actually introns within an exon, as well as intron retentions themselves. All of these things then can lead to non-productive message and protein. As you can see, there are a lot of them. These numbers will actually change as we are constantly generating more data. Obviously, in some cases, they're going to be tissue-specific expression. So we're looking at tissue-specific RNA-seq data as well. And then the world outside is continually working as well, and we're mining that data. So these numbers, but basically we're talking about probably 100,000, and we use different cutoffs as well in terms of the evaluation process itself, because you're looking at a few of these events in potentially millions of reads. So we cross-reference that with respect to the number of genetic diseases. We go through this process of assessing various factors and trying to decide what's the best target for us to look at, and then walk through this process, and I'll show you some of the data associated with that. So again, the kind of events we're talking about, and I'll give you some examples of these, are alternative splicing events in which there could be an exon exclusion or an inclusion of an exon, alternative splicing events, and the alternative intronic events. And these are the kind of numbers we would generate for these type of events. And what that leads to then is a substantial number of diseases that we can potentially execute on. So here are some of the examples that we were just talking about on an individual basis, if you will. So these are examples of a cassette exons, alternative splicing sites, and the alternative introns themselves. And the diseases are indicated here with respect to the targets, SCN1A, SYNGAP, and in this case, PD-L1. So here's some of the data. And the data that what I'm showing you here is in the presence and absence of cyclohexamide that inhibits translation. And as you can see, in these red bars, again, are the alternative events that causes nonsense-mediated decay itself. As you can see, in the context of cyclohexamide, this band increases substantially. Now, in the context of SYNGAP, we actually don't see much of a change. But as you can see, this alternative event is actually quite robust itself already. And then similarly, again, with cyclohexamide, we see this event. This is a secondary event as well. And by qPCR, and we can give a semi-quantitative analysis of how much we can actually generate. And again, I'll show you some data there as well. And this is just to, again, looking at those three examples. What the data here is that we've identified specific ASOs that can target those areas. And these are dose responses. Events, here's 30, 60, 120 nanomolar. And so on the right-hand side are the ASOs with increasing concentrations. And what you can see is that, for example, with SYNGAP, there's in fact a decrease in the NMD event itself, increase in the productive event via TACPA PCR, and an increase in the actual protein itself. So that's our key process going forward, essentially. And we see the same thing with CD274, PD-L1, as well as SCN1A, which I'll tell you a lot more about. Okay? So we are essentially an alternative to gene therapy. We talked about gene therapy approaches, right? So what are some of the differences? Well, before I talk about the differences, one of the key things certainly is that that this is really applicable to a variety of loss of function diseases because we're really targeting the wild type itself. I just want to re-emphasize that actually. So the other thing is the ability to utilize this across small and large gene targets. We're using antisense oligonucleotides, looking at the endogenous expression, so we're not really impacted by how small or how large the transgene needs to be itself. Whether it's a 3 kb or 12 kb gene, it doesn't matter to us, basically, okay? And then this no observed unwanted off-target effect is the idea that we're only targeting the endogenous pool of message. So if our ASO get into a cell type that is not expressing the message, you're not going to get an increase in the productive message itself. So we're using the endogenous promoter system or the endogenous regulatory system to drive that process, okay? And that's very different, obviously, from the challenges from a gene therapy approach, where you have to identify a promoter or elements that's ideally expressed in your target cell itself. And that's one of the key differences as well. But with that, obviously, it also comes to the idea that there's some things we can't do. We can't generally address recessive diseases because if it's not there, it's not going to work. We can't really address gain of function mutations. That's one of the things I think that would be very interesting to talk about because I think that's really crucial for us in that context, is that obviously you have a gain of function mutation, we increase that, that's an issue for us as well. So there's a lot of advantages, but there's potentially some things we can't get at as well. So we focus on haploinsufficient diseases, hypomorphs. We can do pathways. The PD-L1 is obviously a pathway where we can look at autoimmune diseases of various types. There are other pathways that we're very, very interested in as well. But in terms of the short-term, near-term activities, it's really about our ability to address haploinsufficiency diseases or hypomorphs, essentially. There's some other things that we can do, obviously, with ASOs. We can titrate the ASO and get the level of expression that we really want, and that's one of the things we do in our IND-enabling processes. And then we can utilize this across a wide array of diseases. As we were just talking about with Spinraza, there's proof of concept in terms of the ASOs getting into the CNS space. We know we have really nice data in the eye as well, where just by intravitreal injection, we get to the back of the eye and get to all the different cell types in the back of the eye and drive activity. And obviously, manufacturing is not a small matter. I think the people in the gene therapy, I was in the gene therapy world for, I guess, kind of still am, for many years. And as the progress in gene therapy has occurred, it's very clear that manufacturing is one of the key issues for these type of therapies, particularly systemic type of therapies. Okay, so how do we do this? I'm going to go through very quickly our lead program, which is in SC1A. Some of you, and probably many of you, have seen some of this data before. Because of, I guess, we're a public company and embargo, there's some of the new data that I actually can't talk about. They're going to be at the AS meeting on Saturday and Sunday. Most of those presentations where we're going to show some of the non-human primate studies that we've generated, as well as some additional really nice EEG data that we're going to be presenting as well. So what we do is walk across this area of a particular target we've identified. In this case, inclusion of this exon leads to a frame shift and a stop codon in the next exon, and that leads to early termination of that message with respect to the translation process itself, okay? And as you can see, when we walk through this process, there are many that really doesn't change at all, and with respect to the controls. So this is a control, these two are control lanes, and some are very similar to that. There are also places where there seems to be a general knockdown itself, but clearly there are places where the top band is essentially gone, but there's an increase in the lower band. And this is a really nice example. And associated with that, then you see this decrease in NMD and an increase in the productive message itself. So that's really the way we make the sausages, if you will. So very quickly then, we did a series of experiments. We moved in vivo, and as you can see, we see this activity exactly the same way in an in vivo environment, where from 0.3 to 10 micrograms per mouse ICV injections, we see a decrease in that non-productive message, an increase in productive message, and an increase in protein. Specificity. One of the things, obviously, with SCNs is that this is a family of genes that's highly homologous. We want to make sure that there's really no impact on some of the other channels, and as you can see, this is certainly the case here. And in terms of the selectivity, it really depends on the sequence you're looking at. If there are common sequences for splicing and other things, yes, that's really potentially an issue, and you have to make sure that you look at that. But if your target sequence happens to be one that's relatively unique, and obviously once we have the right sequences, we go and inquire into the database with respect to the selectivity of that process itself. So then we moved into the Dravet mouse model, which is one of the nicer mouse models with respect to replication to the human situation. And as you can, see, in these experiments, we're again injecting one dose at P2 ICV, and then we're looking at this long term. As this part of the process, you can see 7 weeks and 14 weeks later, the amount of NAV 1.1. So we look at message as well, of course, but the most important thing is the protein. So what you see is that that protein level, even 7 weeks and 14 weeks later, has been essentially restored to wild type levels. Now we do see an increase in the wild type animals as well. Interestingly, with the enhancement in SC1A, we did not see any negative connotations associated with that. And associated with that, this is done in collaboration with Lori Isom's lab, who have been tremendous collaborators. And the data is quite clear. In this model, there's early death, SUDEP, associated with these animals. And with the drug, again, 1 dose at P2, you can see this very nice retention of mortality. So in a quick summary then, we've moved on to non-human primate data. As I said, there'll be a lot more data at the AS meeting on Sunday. But essentially, we've found that these oligos to be quite safe with really not a lot of issues that we've observed. In summary then, that we've shown that we can increase the message and protein levels in vitro and in vivo. We can address the disease in a very relevant disease model. This is a persistent activity. We think that probably 2 to 3 times a year in dosing is sufficient, which is quite doable. And that we obviously have a well-tolerated pharmacologically active dose. Just to a little bit of advertising is that if you want to learn more about some of this activity, we have the presentations on posted on Saturday as well as Sunday with respect to the respective titles, as well as there's a session that we'll be part of in terms of the genetic epilepsies, as well as Lori Isom is going to be presenting in a workshop on Sunday as well. So we've made, we think, very good progress in the genetic epilepsy disease with respect to Dravet. We've learned a lot in that setting. We're learning more as we move forward. And so obviously this is an area that we would like to do more work in. And we know, and this is now, I don't need to remind this audience, but obviously there's a lot of genetic diseases, genetic-based epilepsies in the current setting, and there's still no real therapies for that. So we think that there's a lot of places where we can continue to work on. We are working on a variety of areas. I showed you some data on SYNGAP. We have nice data on CHD2, TSCs, and obviously we're very much interested in SLC681 as well. And so these are things that are some of the ongoing activities. This is a snapshot. We're actually a lot bigger than that now, but we haven't had a more recent picture of the team. This is obviously a team effort and a lot of people contributing to it. And also I want to really highlight the fact that, this is the in vivo studies has been an ongoing and very nice collaboration with Lori Isom's lab at the University of Michigan. With that, I'll pause, I'll stop and take questions. And I have a minute to spare, which is really unusual for me, I can tell you.
Question over here. Oh, sorry.
Yeah.
So actually, I have 2 questions, if you don't mind. So the Stoke 001 oligo that you used in the Dravet mouse model, is that human sequence? And then in a broader perspective, I guess how important will it be for SLC6A1 to have a mouse model or some animal model that recapitulates the human disease in order to test an upregulation approach?
Yeah, great question. So in the context of SC1A, we were pretty lucky in the sense that the sequence and the event is completely conserved between mouse, non-human primates, and human. It's interesting, it's often with some of these events, so yeah, there's my timer. Sorry. So it's interesting that that we do see conservation in a variety of these situations. So whether that means that they're actually somehow part of the developmental process itself, or something else going on in terms of regulation, we don't really know. It's very, frankly, some very fascinating biology in that context. So we were lucky in that sense. It's not always conserved. We have a project that we're working on now. It's in OPA1 for the eye, for example, and it's not conserved in rodents. And there's a beautiful mouse model, but we can't use it. It is conserved in rabbits, however. So in the cases where there's non-conservation, if the event itself is conserved, we can use a different oligonucleotide to address the disease. So that's how it was done with SMN, actually, right? And so in the context of that, it makes it a little more complicated, but we can still move forward, definitely. Yeah. And the second question, I'm sorry, was? Oh, the model. Yes. So I think that, yeah, I mean, so for the project I was just telling you about with OPA1, we're going to go, and this is something Caddy's talked about, right, where you now have a real progress in the iPSC world with respect to differentiation, using patient fibroblasts, and we know that, for example, in another project by another company, they never went in vivo at all. So I think there are alternatives, but it's still really nice to have, obviously, a mouse model. There's some things you can do that you can't do in that iPS differentiation organoid world. Yeah. Yeah.
Yeah. Thank you very much. This is, I mean, obviously amazing progress. I just have I'm very curious how a company like yours approach the following. So, I mean, this is all enlightened, and this is amazing progress, right? So, it's SCM1A is not equal Dravet, right? Because there's familiar hemiplegic migraine, there's GAPS plus, and so on, right? So, I wonder how do you approach that? That there are some patients who have gain-of-function mutations and they have this familiar hemiplegic migraine, and at the same time, the mouse is not identically like the human disease, right? Not only in terms of heterozygous to homozygous, but also in terms of the clinical representation, right? So it's not one-to-one, and I wonder just what does the FDA require, for example, to show that, for example, too much protein would not be harmful, and at the same time, that the mouse is not identical to the human, right? Just out of curiosity.
Yeah, there's a lot in there. I think there are multiple questions in there, actually. Let me try to unpack some of that. In the context of gain of function and loss of function, I think that's critical, certainly for us. Again, for SCN1A, I think the literature and the data that's out there is quite clear. Probably 80+% are all loss of function. And we have a small set of gain of function where clearly are excluding. But, so for our clinical trials and activities, I don't think that's an issue. But there could be a set of missense mutations assuming we're successful and we're treating patients, that we have to figure out how to address with respect to assays or making sure that obviously we're not harming those patients. So I think that's an important part, and I think as been discussed here, the complexities of this is maybe not so simple as simply loss of function versus gain of function. There could be some potentially mixed type of activity. So developing assays to evaluate and understand the function in vitro systems, I think the big issue obviously is that a lot of times you cannot replicate what's really happening in hex cells or in other, I mean, there are assays where you can put some of these things into a cell system and look for the activity, but how faithful that is in the context of the in vivo environment may be different, yeah. So, I'm sorry, and you were going to say something on.
Yeah, very specific question. When you go to non-human primates, are you also specifically overexpressing in the wild type SMY?
Yeah, so we were lucky in that we were able to actually get PD, right, in all the animal models, in rats, in non-human primates, and obviously we are moving forward toward the clinic. So I think that we're comfortable with the data that we've generated so far. And again, I think, like I said, at the ASO we'll show some additional data there. And so this is a case where I think that whatever is happening, we're not seeing any sort of aberrant impacts of the gain of activity in the wild type situation, basically. Yeah.
Yeah, I wonder if you could talk a little bit about your thinking on how you get the oligonucleotide distributed to where it needs to go in the human brain, because the successes so far, and they have been dramatic. In SMA, for example, you're targeting the spinal cord, which is a little bit easier than the brain. You've got the situation where you target a blindness condition where you can inject directly into the vitreous of the eye. But here, you've got to get this oligonucleotide to where it needs to go in the human brain. And the question is, are there specific areas in the brain that you think you need to get for this disease? Do you have to get it everywhere? And then how do you do it?
Yeah, I think great question. We get asked that quite often, particularly by investors. So in the context of the data that's out there itself, is that actually, although the Spinraza targeting is somewhat different in terms of where you need to get to, they did have, unfortunately, some patients that passed early in those trials, and they were able to generate a nice data profile in the terms of the distribution of the ASOs in the brain. We've done that ourselves, obviously, in non-human primates, and we see a somewhat similar profile. And
is this with intrathecal? Are you doing intrathecal? Is this with intrathecal?
Yeah, intrathecal administration. We can get a very good distribution into the brain area. I think I'm not supposed to talk about a lot of that right here, but on the Sunday poster, all of that data will be presented. In the context of overexpression in that setting, we were just discussing that. So again, I think we can get to those places, and we actually are looking for a pharmacodynamic signal in those areas as well. And certainly the thalamo-cortical regions is one of the areas that we want to make sure that we get to.
So thalamo-cortical area is what you're really focusing on.
Yeah.
In terms of your target region.
It's certainly an area that we can clearly achieve. Yes.
Great. Thank you.
And Caddy.
Yeah. Hi. Yeah. So, how do you determine like 2 to 3 months you deliver one time for the drug? So every year you take the interval of deliver a drug 2 to 3 times a year is enough. Did you decide this by stability of ASO?
Yeah. So obviously we don't know. This is based on our preclinical data at this point in time. As I showed you, a single dose in single injection, 14 weeks later in the mouse, we still have essentially almost close to wild type levels. So we're really kind of trying to project that process. And we know that the ASO is still there and likely active. These are phosphorothioate backbones fully. and their stability is very, very high. And remember what this oligo does, right? It's once it gets into the nucleus, it stays there. It's really like it's really a catalytic event. So it binds, changes the profile of the message. It comes off. It doesn't go anywhere. It's stable. It can bind to the next message, and so on and so forth. So that's why we think that there's such a long-term activity associated with the ASO itself. Yeah.
I thought I heard you say that you were doing intracerebral ventricular injections
ICV for
in
the mouse.
for the
Yeah,
mouse.
because we couldn't do IT. It's very difficult to do mouse IT.
Did you still see any hind limb weakness transiently? That's been seen in a lot of ASO therapies in the mouse models where they are trying to do intrathecal, and I'm just curious personally whether that's how much of that's mechanical or actually something to do with ASOs.
That's a great question, and there's a lot of. It's actually at the recent American the talks meeting that was just completed. One of the big CROs has published a poster, and that's going to come out in a paper that describes all of this, particularly in non-human primates. It's a commonly observed event in preclinical models, but there hasn't been a lot of literature associated with it. And so that will be one of the first ones that have come out. And basically, these are very transient activities, and there's full recovery after a few hours, and certainly by the next day. And it is a dose-dependent phenomenon, but there's also a certain amount of that just with intrathecal delivery itself. Yeah.
Gene, for some of the changes, they're quite substantial. You know, we're talking about up to 10-fold. And if we are going after the wild type allele.
Yeah. Okay, I got it. So this is a really interesting thing that's a little harder to understand through because of the data. So SCN1A is developmentally regulated. What's happening is that at P2 and going through time, we have some really nice data there. You see this increase in productive SCN1A and NaV1.1 until it gets to the adult level, right? So you're starting here, you're ending here. What happens is that the NMD event doesn't change. It's the same all the way across. So what happens is that when we are looking at it very early, and this was the case where we're looking at this at the early time points, that ratio between the two events looks 8 to 10-fold. What we're doing really is we're essentially right shifting the curve. We're pushing the expression to a higher level at the earlier phases. When you get to the adult in the later phases, what we're essentially able to do is about a 2-fold or back to close to wild types. So that's why you're seeing the 8-fold in one place, and actually in the adult we're seeing 2-fold.
All right. Well, thank you so much, Gene. We have